This would be a typical bay in a typical molecular biology lab, usually quite untidy, depending on who works here. This apparatus is called electrophoresis. So this is essentially we cast pieces of jelly, rectangles of jelly, and we stand them in some buffer, and we pass electricity through it. And when the electricity goes through the gel, it separates out molecules of different charge and different size. So you can separate molecules and see how they migrate through a gel. And these are the power packs we use to drive those. Of course, everything's expensive, so that's where the need for money comes in science. We have centrifuges, which we use to spin our tubes, again, to maybe separate different components of a cell. If we put our tubes in here, then the heavy molecules will end up at the bottom and the lighter molecules will be at the top, and we can separate those for use later. This is what we call an FPLC. This is a machine for, again, separating molecules, particularly proteins. It consists of a long tube, and we pump our solution in the top if it's got multiple different sorts of molecules in it, and depending on the migration of the different molecules through a resin in that tube, they either come out sooner or later, and we collect the fractions in this fraction collector. So again, separating out different components of a cell or of a reaction. This is where we grow our cells. So in here we have special hoods which we sit at here to protect our cells from the environment. So all our cellular experiments are done inside one of these hoods. We have other machines to count the number of cells we're using, or microscopes just over here to look at our cells to make sure they're healthy, and a bank of plastics which are stocked up daily in which to grow our cells. And these are all very, very expensive to use. And we try and use as little as possible, but still, that's where a lot of the cost goes. Hang on, have I come to the wrong floor? I have, haven't I? We were one floor up. Shows you I stay so much time on the 9th floor, I can't even count the floors. So here we are in the mass spec facility. This is an important technique for identifying molecules in cells or in other types of, in cells and in chemical reactions. You can almost, it's like fingerprinting. You can have a mixture of molecules, and you can detect a fingerprint that tells you whether that molecule is present and how much of it is present in the sample. It's a very, very powerful technique. It's very expensive to buy a mass spectrometer, and you need people who really know how to use it. I didn't, at the beginning, actively go into scientific research. I just found that the science and the way of life that is science, because science isn't a job, it is a way of life, at least research science, I just found that this is what I wanted to do, and I didn't want to do anything else, and I've never really considered doing anything else. I might be good at something else, but I'm never going to find out, because this is what I do.